Welcome back, everyone. I am going over the digestive system quickly with the overview here from this document again, and then I will be doing the flashcards for the digestive system. So far, I read about intracellular and extracellular digestion. There's a difference between them, where intracellular digestion is mainly the oxidation of glucose and lipids, fatty acids per se, to make energy, but extracellular is just like the breakdown of food that goes into our alimentary canal. And we have mechanical and chemical digestion. So mechanical, as the name says it, it's the physical breakdown of food, and we chunk it down into smaller particles. And chemical digestion is basically with the help of amylase, for example, in our saliva, that breaks down sugar, or we have amylase or lipase. So if you want to break down proteins, for example, you have to break down the peptide bonds, so this is called the enzymatic cleavage of chemical bonds of our like peptide bonds that form between the amino acids. Or for example, starches, you have to break down the glycosidic bonds. Now we have also peristalsis, where this is the rhythmic movements, contractions of our gut tube, and rest and digest. So whenever you are digesting, your parasympathetic system is at work, and your sympathetic nervous system shuts down actually. And the pathway for our digestive system goes from the oral cavity to the pharynx, to the esophagus, to our stomach, and then to our small intestine and large intestine, and then our rectum. And so we start from the oral cavity. So oral cavity does the mastication. So mastication starts the mechanical digestion. So mastication is basically the mechanical digestion of food. And then you also have salivary amylase and lipase that start the chemical digestion of food. So food is formed into a bolus and swallowed. Now I wrote here as a question that do proteins get broken down in oral cavity or not, or are they or is it in the stomach where it's the first site that proteins are broken down into? Now food is formed into a bolus and swallowed, and we have pharynx. So what does pharynx do? It basically connects our oral cavity to the stomach, to esophagus, and it connects basically our mouth to esophagus. And there is also the epiglottis, which is part of the pharynx, and this prevents food from entering into larynx. Now, is larynx the same as trachea? We can figure that out later. Is larynx the same as the trachea? Now we have esophagus, which propels food to the stomach using peristalsis. The top third is autonomic. The top third is somatic. So there is somatic control because it's skeletal muscle. Now the middle third is a mix of smooth muscle and skeletal muscle, and the bottom third is smooth muscle, and both of the middle and bottom third are under autonomic control. And then we have stomach. So our stomach is an acidic environment. The pH is very low. It's equal to 2. And then we have 4 parts of the stomach. We have the fundus, and then the body, and then the antrum, and then the pylorus. The enzyme pepsin chemically breaks down proteins. So we should investigate if proteins are start to break down chemically via pepsin, or are they chemically digested before our stomach? Now, there are secretory cells that line the stomach. We have mucus cells, which produce bicarbonate-rich mucus. So now we know that the content in mucus is bicarbonate, which is to protect stomach wall from acid, because the acid is too strong. We want the stomach wall to protect against it. So that's why we have mucus, which is bicarbonate rich. So as we know, bicarbonate is the base against acid, so we kind of want to neutralize it with that. Now we also have chief cells. Chief cells secrete pepsinogen. It is a protease activated by the acidic environment. So when you have an acidic environment, we have a protease that is activated. So we know proteases break down proteins, and chief cells are the ones that secrete pepsinogen. I had this visualized a couple of years ago with as like chief officers, like sheriffs, having their feet on the table and drinking Pepsi. So that way I know chief cells secrete pepsinogen. Now we have parietal cells, which secrete HCl, an intrinsic factor, which is needed for vitamin B12 absorption. So because you have to absorb vitamin B12 in your stomach, your parietal cells secrete HCl and intrinsic factors, INFs. Or we don't want to get this wrong with interferons that we had in the immune system. But for now, we just need to know that intrinsic factors, IFs. So is there an acronym for intrinsic factors like IF? Now parietal means like we have the parietal lobe in our brains too, which is the middle top section of our skull, right? Our brain. Now we have G cells, which is the fourth one, and G cells secrete gastrin. It is a peptide hormone that increases HCl secretion in gastric motility. So G cells, gastrin, it makes sense. What does gastrin do? We have gastrin here. No. What does gastrin do? It's a peptide hormone that increases HCl secretion in gastric motility. So because you need so gastric means does gastric refer to the stomach? So probably gastric refers to the stomach, so that way we know. gastric of the stomach. So exactly, when you say a gastric ulcer or a gastric acid secretion, it refers to the stomach. So when you want to increase gastric motility, you have G cells to secrete gastrin, and gastrin, which is a peptide hormone, also increases HCl. So after processing in the stomach, food particles are now called chyme, however you want to say it. And chyme exists through pyloric sphincter, which then attaches to the duodenum. So chyme exists through pyloric sphincter. We had the pylorus, which is the last section of our stomach. So we have the pyloric sphincter, which then transitions into duodenum. We will see more of this, its anatomy, probably in our flashcards. If not, we can search for it. Now you see here that there are feeding behavior hormones. There is 6 of them here, and they go 2 by 2. So we have ADH and aldosterone. If you remember, they both increased or decreased blood pressure. They increased blood pressure. And so the increase in blood pressure results in increase in thirst as well. So what hormones function on our hunger? We have glucagon and ghrelin. So when you have glucagon and ghrelin, when you secrete them, you go grr, you go hungry. You also have leptin and cholecystokinin. So leptin and cholecystokinin increase satiety, and when you're satiated, it means that you're not hungry, right? So there's a way that people lose weight. Nowadays it's with Ozempic or Mounjaro, and the main hormone that it has an effect is glucagon-like peptides. So what does this glucagon-like peptide do? It mimics glucagon. So glucagon is the reverse of insulin. So when you want to uptake the sugar by your tissues, you need insulin, right? When the glucose is needed to be uptaken, you need insulin. But when you want to get rid of the glucose you have stored in your body, for example in your liver, which is mainly glycogen, you can use glucagon to get rid of the glucose there. So it takes away from your glucose, and therefore you can get hungry, right? As a result, because your body is getting rid of the glucose or the sugar. That way you don't have any more sugar or glucose, so you get more hungry. But we can still ask, how does glucagon or ghrelin increase hunger? How does ADH, aldosterone increase thirst? And how does leptin and cholecystokinin increase satiety? Now we have duodenum. So duodenum is the first part of small intestine. So it's a basic environment. It has a pH of 8.5, and it's a site of the majority of chemical digestion. So the majority of your chemical digestion takes place here at the duodenum. That's very important. And the enzymes in duodenum are disaccharidases, aminopeptidase, and dipeptidase. That's for your proteins, and enteropeptidase. So what does enteropeptidase do? It activates trypsinogen and procarboxypeptidases. So we need to, first thing first, we should not confuse trypsinogen with pepsinogen that we had here. So it was the chief cells that secreted pepsinogens, but enteropeptidases activate trypsinogen and procarboxypeptidases. We have aminopeptidase and dipeptidase. They are brush border peptidases. So I believe this is referring to the brush cells in our digestive tract. We have disaccharides. They are also brush border enzymes that break down maltose, isomaltose, lactose, and sucrose into monosaccharides. So as you know, maltose, this should imply that maltose, isomaltose, lactose, and sucrose are disaccharides. And we have secretin. So these are hormones in duodenum. We have secretin and cholecystokinin. We had cholecystokinin there. So cholecystokinin, chole comes from our bile. So stimulates bile release from gallbladder. Chole comes from gallbladder. Anything related to like cholesterol or chole refers to gallbladder, and it stimulates bile release from gallbladder, release of pancreatic juices, and satiety. So it stimulates satiety because it releases the bile from gallbladder. Now we have secretin. It's a peptide hormone, and it stimulates the release of pancreatic juices and slows motility. So we had something that increased gastric motility, which was gastrin from G cells, right? Now this is reverse. Secretin is the opposite of gastrin, where it slows motility. Now, does secretin slow the motility of the stomach or the small intestine? Okay. Now we have absorption and defecation. I think defecation is pooping. So absorption first. The jejunum and ileum of the small intestine. Those are the second third and third third of the last third of the small intestine. Jejunum and ileum. They are primarily involved in absorption. So now we know that the majority of digestion, chemical digestion, happened in duodenum, but then now we're talking about absorption. So digestion is basically the breakdown of the food, the energy molecules, but absorption is getting the energy from them, right? Getting the nutrients from them. And jejunum and ileum are primarily involved in that. And the small intestine is lined with villi, which are covered with microvilli. So this is like a matrix thing, which is crazy. So your small intestine cells have these villi on them, and those villi also have microvilli. We'll see pictures of that. So villi are in capillary beds, and if you remember, this was in the circulatory system that we studied about. So the capillary beds absorb water-soluble nutrients. So yes, because our blood is with water, and everything water-soluble can be absorbed in the capillary beds. And there's also lacteal type of villi, which absorb fat and they send it to lymphatic system. So we have both lacteal and capillary bed. Are villi two types? Capillary bed and what was it for the lymphatic? The fat soluble? Lacteal. They absorb fat sent to lymphatic system. And lacteal. We have vitamin absorption, so it's fat soluble. I know KADE are fat soluble, and they enter the lacteal. And water solubles like B and C, they enter plasma directly. Now we have the large intestine, which absorbs water and salts. It forms also feces. We have cecum. It's the outpocketing that accepts fluid from small intestine through ileocecal valve. So cecum is the first section of large intestine that attaches to the ileum. It's a site of attachment of the appendix. So our appendix is also attached to the cecum. The structure of colon is first ascending, then transverse, then descending, and then sigmoid. And the gut bacteria, they produce vitamin K and biotin, which is vitamin B7. Do the gut bacteria live in the cecum of the large intestine only? And why do they produce vitamin K and biotin B7? And is that enough for the body? Now we have accessory organs, which is the last part. They originate from endoderm. That's the developmental information that we might need to know for the MCAT. So the accessory organs originate from endoderm, and they consist of pancreas, liver, and gallbladder. So pancreas has acinar cells which produce pancreatic juices that contain bicarbonate, pancreatic amylase, pancreatic peptidases, and pancreatic lipase. And we have liver, which synthesizes bile, albumin, and clotting factors. It processes also nutrients. It also does a detoxification from ammonia to urea, as well as alcohol and drugs. That's very known for that. And liver receives blood from the abdominal portion of digestive tract via hepatic portal vein. That's also very interesting. I need to dig more into that. I remember this kind of from the days of my anatomy classes. And then we also have gallbladder, which stores and concentrates bile. And what bile we know it's used for fat, digesting fats. So CCK, cholecystokinin, stimulates bile release into biliary tree, which merges with pancreatic duct. Okay, cool. I will dig more into this. I might have to change my location, so for now, I'll be right back.